Okay, hi everybody. We're ready to start chapter 13, entitled Microbe-Human Interactions, Infection, Disease, and Epidemiology. So one thing that we have to keep in mind as we talk about microorganisms and the role they play in influencing our health is the fact that we are constantly living in a state of homeostasis. We sort of underlined and emphasized that concept back in A&P. What we didn't talk much about at that time is the role that microorganisms, bacteria, fungi, some protozoa play in terms of both benefiting and sometimes harming us. And so there is a constant give and take, if you will, between us and these microorganisms. And we're going to talk about both the good and the bad aspects of that symbiosis here in chapter 13. And I think the book does a really nice job sort of giving three significant bullets there at the beginning of this chapter in this section 13.1, We Are Not Alone. When they talk about the fact that microbes provide a protective and stabilization effect on body surfaces by establishing themselves as normal residents, that they're involved in the maturation of host defenses and in the development of the immune system. It's not something we talk too much about, but it's very important. And finally, the fact that they do sometimes invade and grow in and on our tissues where they can cause damage. So we're going to talk about a whole host of different interactions that these microorganisms have on you and me. And we want to begin, as we often do, by defining some terms. So when we talk about resident flora or microbiota, we're talking about those that live in and on us. And typically they do so in a mutualistic or commensalistic way. So if you think back, I think we defined mutualism in an earlier chapter. Mutualism is that sort of intimate symbiosis in which both parties benefit from the relationship. Commensalism, as I've noted here, really gets to the idea that there's this peaceful coexistence that takes place. We're not harming each other, we're not hurting each other, nor are we really necessarily benefiting from one another. We're in this sort of neutral zone, if you will, a peaceful coexistence. Not mutualism, not parasitism, somewhere in between. If you run across the term indigenous flora or indigenous microbiota, the term, as you see here, just simply means belonging to or native to the organism, which would of course be us, the indigenous bacteria, I should say, not us, but the indigenous microflora are those that belong to us, live on us, live in us. They're indigenous, they're native. Infection, pathogen, infectious disease, kind of, I think, are terms that we can all get a sense of, so I'll let you just kind of read that over. Residents are those that become established in and on us, usually at the moment of birth or during the birthing process, or shortly afterwards. We begin to acquire bacteria. Some of those will become established, called residents, as opposed to some that may only appear or occupy our body surface or become part of our internal digestive system, let's say, or respiratory system. They're not there for long, they're transients. And sometimes transients can cause problems. But typically they're not there on a long-term sort of basis. So when we think about where we would look for microorganisms, and I guess when I use that term, I'm focusing primarily on bacteria, But we do want to keep in mind that earlier in the semester, we said microbes could be bacteria, they could be viruses, which are of course acellular, they're not living. They could be protozoa, they could be fungi, they could even be helminth parasites like roundworms and tapeworms and things of that sort. So we're going to kind of again remember microbes, primarily bacteria, but let's not forget the viruses and the protozoa and fungi. We'll focus on those a little bit in this chapter, mostly bacteria. So where do we find these normal resident microbes? Well, you can see from this table that the surface of the body, those areas that have a portal of entry, right? Those that can get in from the outside in some way will be harbored in those parts of our body that have those openings, basically. So it kind of makes sense if you just think about it. When you look at internal structures, organs and tissues, or fluids within organs or tissues, we find a fairly sterile field. Not fairly, we find a sterile field. So when you look at the heart, when you look at the circulatory system, when you look at bones, when you look at lungs, when you look at skeletal smooth muscle, cardiac muscle, kidneys, bladder, our blood, our cerebrospinal fluid that circulates, these are all sterile fields, sterile fluids, sterile tissues. Now we know that our resident microbiota does change over our lifespan. While there's some consistency between the type of bacteria or microbes that you find, let's say when you're 15 versus when you're 85, there's going to be some bacteria that you're going to find, in those two relatively different age groups. But as this slide sort of indicates, the population of microbes in and on us can change through our lives, depending upon our health, what kind of foods we eat. Hormonal shifts can significantly impact the microbiota, especially in females. The types of drugs we're taking, as we know, can interrupt the normal residence and allow transients to gain a foothold and cause problems. We talked a little bit about that in the preceding chapter with regard to superinfections. Remember that? And this second bullet is just sort of interesting. We take it so for granted, the fact that we shower every day or every other day. Is that something we should be doing? Does that promote our health? Well, it maintains our hygiene, one could argue, but there's some studies, as I've said here, or make note of here, that question whether daily showering may actually not be healthy for us because it might prevent the normal flora from building to suitable levels to provide us with increased health. Interesting. So when we consider those residents that provide us with beneficial health aspects, they do so by preventing transients from gaining a foothold and causing problems. So we often talk about this microbial antagonism that exists, where the good, quote unquote, microbes are fighting against the intruder or transient microbes. And that battle is being waged 24/7, 365. But we know sometimes the transient can gain a foothold again if we become sick, if our immune system becomes compromised, if we suddenly change our diet, if we have other chronic or acute health issues that come about, that all can influence the microbial balance and affect lots of aspects of our health. When we consider the fetus and the newborn and follow what happens as development occurs in the uterus and shortly after birth. There's some amazing things that take place with regard to what sorts of bacteria get inoculated onto and into that newborn. For the most part, during fetal development, the fetus is in a fairly sterile field for the most part. But during the moment of childbirth, as these amniotic fluids begin to leak out, and when we start to tear some of those membranes, then we interrupt that sterile field. And so from that very moment when the amniotic sac ruptures prior to birth, that could be several hours before, or it could be literally minutes before, we now know that there can be and there is introduction of bacteria from the mother's reproductive tract to the fetus, or to, in this case, the newborn, as it's being passed through the birth canal, as we see here. And then from that moment on, once that child is exposed to the environment coming out of mom, whether again that's C-section or normal vaginal birth, that process of inoculation begins and continues really for the entire life of that individual, ranging from interaction with mom or dad or the nurse or other individuals, to the type of milk that's being provided. Is it a formula? Is it breast milk that mom's providing? Your book talks a lot about this. Make sure you look it over. The benefits of breastfeeding far outweigh the disadvantages of that process, although it is can be a difficult thing for a new mom to do sometimes. But there are some super positive benefits from breastfeeding, providing a more natural inoculation of the baby's digestive tract, less GI complications, like less colic maybe for the newborn, but also giving that newborn a real positive start with respect to how its immune system develops. So please make sure you read that section over carefully. I think you'll find it very interesting. So where do we find these indigenous microbes? Well, as we've said, generally the surface of our body and any sorts of portals that would introduce bacteria from the outside inward. So this table is really an excellent summary that you can come back to if you ever want to get a quick glance of the types of microbes. And we're talking here about mostly bacteria, fungi, and protozoa, or maybe even some arthropods like mites in the skin. It's kind of interesting, kind of creepy, but interesting. You can come back to this table and also note that there are some commentaries, some remarks given for each of these 2, 4, 6, 7, 8 different regions of the body. What we're going to do next is we're going to talk a little bit more in detail about these different anatomical regions. Before we do that, I do want to make mention that I have posted a newspaper article that I scanned and I placed in the Blackboard course shell. This is about, I think, maybe 4 or 5 years old. Just a short little 3-4 paragraph article. Make sure you check it out. Kind of interesting. I should have probably put this in chapter 12 PowerPoint, but since we're talking about the human body, and we'll be getting into the respiratory system in just a moment and some of the bacteria that we find there and so forth, I thought I'd throw it in here. So just take a look at it. Just a quick overview comment with respect to viruses. We really will not be talking a whole lot about viruses so much in this chapter, a little bit, but not a whole lot. And I think it's really a topic that the book should spend more time talking about, and that is, what role do viruses play as part of our normal microbiota? Now, we know that they're acellular, they're non-living, we often associate viruses with disease. And I think we need to also understand that there can be beneficial aspects of viruses too. And the book just doesn't go into a whole lot of detail about this, other than to make some mention of these endogenous retroviruses. Now, you remember what retroviruses are. HIV is a retrovirus. It contains that reverse transcriptase. I'm not going to rehash all that. But it's interesting. The second bullet here basically says that upwards of 10% of genetic material in you and I may consist of these ERVs, endogenous retroviruses. So we have yet to learn the contributions that viruses play in terms of our health and the maintenance of homeostasis. But I think as we learn more, we'll come to understand that there are some real positive benefits that viruses can provide us, even though we talk about the negative aspect of viruses or microbes in general more than we talk about the benefits. In your textbook is an interesting making connections section that talks about the human microbiome. And in essence, what they're doing is they're surveying different parts of the body, obviously in this diagram, both posterior and anterior views. And they are giving us some semblance of where certain families of bacteria might be harbored. And they're color coding these. So as you peruse down, the list of locations, you'll also see some common colors. Now, again, I'm not expecting you to know this for a quiz or an exam, but we've got, at least 10,000 different species of microorganisms that make their home in and on us. That's just mind-boggling. But you might remember back to that TED Talk that was given in one of the earlier chapters, where the presenter introduced the concept of just how many prokaryotic cells we have in us and on us compared to how many eukaryotic cells make us up. And it's an exponential difference. We have many, many more prokaryotic cells in and on us, number-wise, than we have eukaryotic cells that make us up. So it's just kind of interesting when you think about it that way. So let's talk about some different parts of the body, starting with the skin, and make some observations as to where we would find these microbes and some commentary about them. Well, as we know, the skin is the largest organ system of our body, the integumentary system. We have both residents and we have transients, with the transients being more impacted by our hygiene. And this gets back to the idea of, should we be showering or washing every single day? And is that somehow maybe affecting the normal balance between transients and residents? And so what the book has adopted is sort of a yellow coloring schematic here to show where you'd expect to find microbes. And so as we look at the diagram, we see, of course, the epidermis. We see some regions here in the sweat glands and the ducts that lead from the surface, or from the sweat gland, I should say, to the surface. These, of course, are sebaceous glands, which you remember are also open to the surface. So generally, we would not find any microbes in the dermis, here in white, or the subcutaneous layer or hypodermis, shown here sort of in gray. These are, of course, adipose cells. Staphylococcus, Corynebacterium, different types of yeasts are the common residents that we tend to find on our skin. And again, I think those of you that have had lab or are taking lab, you know about Staphylococcus. There are numerous species of Staphylococcus, but some of these are normal. They're good. They're beneficial. They're not going to cause us infection, typically, unless maybe we break the barrier here and introduce those down into the dermis or deeper tissues. But as they exist here on the surface, they're not causing really any major problems. The gastrointestinal tract, which you recall from A&P, is really from the oral cavity all the way down through the esophagus, stomach, small intestine, large intestine, rectum, and then finally to the anus. We're going to talk next about the oral cavity and upper GI tract, and then we'll focus a little bit on the large intestine and small intestine. We won't talk a whole lot about the stomach. That doesn't mean that there aren't bacteria that live there, because there are, but there's not a great diversity of microbes in the stomach as compared, let's say, to the small and large intestine. There's just no comparison. There are many, many more diverse species in these areas of yellow than you would find, for example, in the stomach. And the reason for that, of course, is because the stomach has that very, very acidic pH of about 2, right? That's going to really prevent many, many bacteria from surviving in that sort of inhospitable environment. There are some that do, that love it. You know, they love acid, but most bacteria don't. So again, think about the fact that we have an opening here at the mouth, and we have an opening here at the anus, the rectum. So this is, in some sense, sort of a closed, yet somewhat open tube, continuous with the outside, right? So the mouth. Some studies suggest that we have over 600 different species of bacteria and microbes living in our oral cavity. That's just absolutely astounding when you stop to think about it. Maybe you would have said a couple dozen, maybe 100. No, we're talking potentially over 600 different species. And when you look at a milliliter of saliva, which is about 20 drops, we're talking about 5 times 10 to the 9th number of cells. So that's 5 with 9 zeros after it, right? That's a lot of bacteria. Streptococcus is a common genera that we tend to find here. And several species of strep, particularly mutans and sanguis, as I've noted here, contribute to cavities. Dental caries are cavities. And these 2 particular species of staph form this, or strep, excuse me, not staph, streptococcus, form the sticky slime layer on the surface of our enamel due to the sugars that are present in our mouth from our food. And the adherence of those sugars to the tooth enamel provides the perfect condition for a biofilm to be produced. And we talked about biofilms and the complexity of those structures. Yeah. And it's the production of acids that the biofilms produce that basically can decay the enamel and cause holes in the enamel. And that's what a dental carie basically is. It's due to the acids produced in those biofilms or by those biofilms. And you probably have always heard, or maybe you haven't, this notion that it's better to be bitten by a dog than to be bitten by a human, because humans harbor many more potential pathogenic bacteria should they get into the bloodstream than even a dog might, which is kind of bizarre, but that's what they say. But any of you in the healthcare industry do know that human bites can be very, very bad. They can cause major, major infections. As we work our way now down into the lower GI tract, and of course we're here in the large intestine, it's here that we find lots of anaerobic bacteria or some aerotolerant bacteria, but it's a pretty anaerobic environment for the most part. The number of microbes here in the large intestine probably reaches its peak when you look at the entire GI tract as a whole, with the number of microbes in feces reaching upwards of 10 to the 11th power number of cells per gram. So upwards of 30% by volume of feces is bacteria. Yeah, not to get disgusting, but that's pretty much what we're talking about. With respect to the appendix, which you know is at the distal end here of the cecum, just before we encounter the ileum. So coming off of the small intestine, where, not the distal part, the proximal part, I should say, of the large intestine, where these two meet is this ileocecal valve, which regulates movement of materials from the small intestine into the large intestine. And here's the appendix, okay? We often talk about the appendix as it relates to appendicitis, inflammation of the appendix, but there's been some studies done by Duke University to suggest that the appendix may have another role to play, and that is in helping to inoculate the colon, here, the large intestine, with beneficial bacteria, especially after there's been a bout of diarrhea and you've lost a lot of those, you've flushed a lot of those beneficial microbes out. The appendix, while we know it's also lymphatic tissue, may also play a role in terms of the microbiology of our gut, of our large intestine. So I thought that was kind of interesting. Going back to feces for just a minute, not to get gross about it, but the book talks a little bit about how those bacteria that are fermenting these waste products produced by other bacteria can produce important vitamins that we would not get from our food. So B12, riboflavin, thiamine, yes, you can get those from food, I guess, but the point being, you don't have to ingest food with those vitamins, because microbes in our gut, in our large intestine, will produce those as a natural process, and then we absorb those important vitamins. And in addition to the vitamins, your book also talks about different types of beneficial acids are produced here as well. They mention acetic acid, butyric acid, propionic acid, manufactured by microbes. I'd like you to watch this 8-minute YouTube video. It's a very interesting video that talks a little bit about how people who have had major issues with regard to being unable to digest food properly sometimes have to have or resort to fecal transplants. So check that video out. Very, very interesting. Okay, moving away from the skin and from the GI tract, let's now talk about the respiratory tract. Certainly we know, as we said a moment ago, that our mouth harbors a lot of bacteria. But as we move down toward the upper parts of the respiratory tract, we find bacteria like Staphylococcus aureus. Again, those of you in lab, you've heard of this. These are natural residents in our nasal vestibule. This is that region here, and in the anterior nasopharynx. Remember, nasopharynx, oropharynx, laryngopharynx. So all these areas shown here in yellow harbor bacteria. Neisseria is another common genera of bacteria in the mucous membranes here in the nasopharynx region. The tonsils here in the naso and oropharynx can harbor Haemophilus, the genus Haemophilus. Once we get basically beyond the larynx and the upper part of the respiratory tree, once we get, into the tree itself and into the lungs, we're pretty much dealing with a sterile field. Our immune system and the presence of phagocytic macrophages are going to phagocytize any possible bacteria that might get beyond this region or fungi. Microbiota of the genitourinary tract, so we're combining both urinary and reproductive systems here. Again, note the areas in yellow with regard to the female, the vagina, the external genitalia, upper, or not upper, but distal regions here of the urethra may harbor microbes. And then the uterus, for the most part, pretty sterile. Cervix would be the extent to which the bacteria and microbes probably would get not much beyond that. And in the case of the male, of course, the scrotal sac, being part of the integumentary system, is going to harbor bacteria. The glans penis, the foreskin, if it's an uncircumcised male, and just a little bit of the urethra here. Once we get beyond the first centimeter or two of the urethra, from there back up into the bladder, sterile for the most part. Now, we've talked a little bit about this in the preceding chapter with regard to superinfection, but I think it's worth noting again how in the vagina we have those residents that are keeping the vagina fairly acidic. So in that microbial antagonism, keeping those yeast transients sort of in check for the most part, because these lactobacilli that are normally there are metabolizing and fermenting the sugars. And whenever you ferment sugars, you're going to get acids produced. You remember that from lab probably. And that drop in pH down to 4.5, it's what is what's keeping the Candida albicans yeast in check. But if a woman is on an antibiotic regimen because of another infection, as we said in chapter 12, that can throw off the pH. You may end up killing the lactobacilli indirectly, causing Candida to gain a foothold, and then you're dealing with a yeast infection. So it's a superinfection, comes about as a result of antibiotics that have been prescribed for a different infection somewhere else in the body. So how do we maintain our normal microbiota? Well, certainly we could say that the foods we eat, our overall health, is critical in the maintenance of a homeostatic sort of situation with respect to bacteria and other microorganisms. And having a healthy immune system is enhanced by having a healthy microbiota. So the foods we eat, our overall health, lots of factors can influence this, as we've talked about earlier. What's been gaining a lot of press, if you will, in the last 30 years, 20, 30 years, is this idea of people taking lots of probiotics. These can range from different kinds of yogurts to sauerkraut to different kinds of drinks or different types of cheeses. These can all sort of re-inoculate the body. So when somebody, let's say, who's had a disease or diarrhea, for example, I think everybody knows that yogurt can be a really good way of inoculating the body with microorganisms that might have been flushed out of the system due to the diarrhea. But a lot of people are taking these probiotics, each and every day, irrespective of being sick, with the idea that they're promoting the healthy balance between good bacteria and bad bacteria. And this photograph was taken from a Wegman store in Jamestown a couple of years ago. But I'm sure you can go to any grocery store, or Walmart. Some of these larger convenience stores are devoted to a whole bunch of different probiotics, and some of you may actually even be taking them regularly. Okay, I think I'm going to stop this first lecture in chapter 13. And when we get together then in the second one, we'll talk about factors at play in the development of infection.